The views expressed on Science for the People are not necessarily the views of this station, its affiliates, sponsors, or advertisers.
Hello and welcome to Science for the People. I'm your host, Desiree Schell. This week we'll be talking about disease from two different angles. How do we investigate a mysterious epidemic? And how do we go about making our own, if we wanted to? A little later, we'll be speaking to Benjamin Miner about starfish wasting syndrome, and we'll be throwing in a brief interview with Ross Lockwood about the high seas Mars simulation, just for fun. But let's kick it off with how and why we create brand new diseases. Hello and welcome to Science for the People. I'm Desiree Schell. I'm here with Dr. Michael Osterholm, Director of the Center for Infectious Disease Research and Policy and Director of the NIH-supported Center of Excellence for Influenza Research and Surveillance. He's also a professor in the Division of Environmental Health Sciences, School of Public Health, and an adjunct professor in the medical school at the University of Minnesota. Welcome to the show, Michael.
Thank you.
Now, in previous episodes, we have spoken about virology and vaccines and such, and I think it's well established by this time that this is a necessary and vital field. But what we haven't spoken about is creating new diseases on purpose. So why might one want to do that?
Well, first of all, actually the concept of creating new diseases or taking old diseases and making them do harm to people on an intentional basis in itself is not new. We know that dating way back to the Middle Ages, that when there were wars going on, that they would basically hurl plague-infested with flea-loaded bodies that were dead over the walls of various cities, trying to, in fact, start plague behind those walls. And so this is not new. And what we realize is that over the past several years, we've taken this to potentially a whole new level, though, of where someone could do something very, very devious with microorganisms because of our ability today to genetically change them in ways that we could never do before. So I think we've really hit a sea change in the idea that someone may want to do harm with these bugs. In addition, today, when you're working with these particular organisms, the potential for actually not intending to do harm, but doing research with an organism that you alter in such a way that is now much more transmissible or it can kill with much more efficiency and accidentally escape from your laboratory is a reality that we had not, in fact, really considered until just very recently.
Well, there definitely are researchers that are doing the same thing with the best of intentions, correct?
Right, and that's one of the things that has made this so difficult today in terms of a point of scientific discussion and the public's right to be involved with that scientific research from at least a policy perspective is that we have scientists working with organisms today where their work is really intended to actually help society by better understanding how that particular organism may gain the various factors or characteristics that would make it more efficient, for example, for transmission, or that might actually kill with more efficiency. And so in doing that work, the intention is absolutely, yes, let's see what we can do to help society. The problem with that is if that information then is detailed and it is made available to the public, it basically allows anyone to now, in a much more easily obtained way, do that kind of work. And so that's one thing. The second thing is, again, it's just can the virus actually escape from the laboratory where it's at just by accident? We have laboratory accidents that happen with some fair frequency, and including accidents in some of our most secure laboratories. So those two factors now must be weighed against the issue of what is the good from this versus what is the potential harm that can occur with this, and how do we decide that? Who decides that? And do we have ways in which we can take that standard and apply it globally, not just in a few countries, but across the world?
Well, let's start with the science, and then we'll get into the ethics. So what kind of pathogens are we talking about then?
Well, you can do this with any pathogen, and a pathogen being a disease-causing organism. You could make a common foodborne pathogen much more lethal by, in fact, inserting now into it the kind of changes genetically that now makes it capable of producing toxins or poisons that it wouldn't have done before, or in fact the fact that the infectious dose may be much lower because of certain protective factors of the bacteria now that allow it to escape some of the damage that the acid of the gut does and so forth. So there's something like a foodborne pathogen. On the other hand, you have those agents that have been traditionally associated with bioterrorism or biowarfare in the past. Anthrax, tularemia, plague, and even, God forbid, smallpox. Those are the kind of organisms where we should never really be doing research there as it relates to just helping the general public with everyday disease, because in most cases those diseases aren't everyday disease problems. Clearly, Bacillus anthracis are the cause of anthrax, or the causative agent of plague are naturally occurring, and we do see some cases, but what we're talking here is a kind of research that would really heighten their ability to kill, and if it were disseminated over a large area, say, for example, from an aerosol device. Then you have the third category, which I'd call really the very legitimate non-traditional bioweapon agents, but ones that surely can cause great havoc in society. Influenza is the classic example, and that's the one that we've really been most frequently confronted with over the past 2 years in terms of this kind of work. Here is a situation where no one had ever thought of weaponizing it, no one ever thought of somehow making it do harm, but the research that is looking at how might it be transmissible from human to human, and what would the changes need to be for a bird flu virus to basically become a human-to-human transmitted pathogen. That's the work that's really concerning, because in fact, while that may give us a clue towards, yes, this virus is evolving towards one that could be readily transmissible between humans, that's also the same virus if that information is published, or if that virus should escape accidentally from a lab, that could raise a great deal of concern from a public health standpoint of what it could do.
You mentioned lab safety earlier. What sort of are the situations around that?
Well, first of all, this is one of those conundrums where we often raise the issue about lab safety in some of the best labs in the world that are doing this work. And those aren't the labs that I'm nearly as concerned about. Where there, they actually take great pains to be sure that this virus does not escape or that it does not infect the workers that are actually working with it. And so from that standpoint, we can really focus on just a few laboratories and make sure that we really have everything the way we need it to prevent any of those viruses from escaping. The problem with that, though, is that if you didn't publish that work, and that's been what you might say the real point of contention has been, is as in science, one of the ways we further science is to make information available so others can have access to it and can reproduce that work and use it to further their own work. But now if we publish that work, we basically, in many cases, will make laboratories around the world that may not have nearly the same detail towards preventing escape from actually occurring. And no one's going to be in a place to necessarily police that. And so one of the things we worry about is this generally made available information that now has a laboratory in God knows where working on this virus for whatever purposes, and that escapes from there. So it's not just the lab safety of those labs working on it currently doing this pioneering work, but it's also all the enabling materials that are out there that now allow labs that could never really have done this work to begin with from doing it. And those are the labs I most fear where this virus could escape from.
Now, this is not an abstract argument, really. I believe in 2012, there was a group doing some controversial studies on the H7N9 bird virus. Can you give us some background there? Because you definitely spoke publicly about it.
Well, actually, it goes back even further to work that I was involved with as a member of the National Science Advisory Board on Biosecurity, the U.S. federal government-based group that tries to bring a certain discipline to understanding how microorganisms could be altered to, in fact, inflict more harm, what we call dual-use research, just by trying to learn more about them, and in how work supported by the U.S. federal government might, in fact, do what it can to prevent those organisms getting into the hands of either bad guys or accidentally being used in laboratories that then have released And so the work really focused first on H5N1, kind of the original bird flu concept virus that emerged in Asia back in 1997, reappeared in the 2003-2004 time period, and continues to be a concern. And what happened was two different research groups, one in Europe and one in the United States, started doing work on these viruses, passing them through ferrets, an animal model that is close to, or at least approximates, as occurring in mammals in general, including humans. And what they were looking for was there changes in those viruses with successive passages indicating that they were becoming basically more accommodating or more likely to be transmitted to a human and then by a human to another human or another mammal. And then by documenting those changes, could they in fact just genetically alter the virus without even having to pass it through ferrets anymore to make that more likely to be transmissible? But sure enough, their work suggested that in fact is what was happening. And this came before the National Science Advisory Board, and after almost 6 months of pretty intensive debate, it was decided that in fact they should be able to publish this work with great reservation. One group received general approval of the NSABB, or the National Science Advisory Board, and the other work got a split vote decision. That started the whole debate with flu virus, because in fact the last thing we need is to enhance the potential for H5N1 either accidentally be released from a lab or someone now working on it who could basically have the expertise before and then for whatever purpose is intentionally releasing it. That then carried over into the H7N9, the current situation we're dealing with in China, which now and together with H5N1 really pose some real challenges for us in terms of potential for influenza becoming a very serious public health problem with a potential pandemic situation if either one of these viruses ever developed the ability to be transmitted human to human. And it was at that time that when H7N9 work first came up this past year, I just reminded everyone that we've been dealing with this for the past 2.5 years, and with H5N1, and I had made a statement back in 2012 saying that the NSABB really never dealt with the issue. We just kicked the can down the road, said, well, in this particular instance, okay, go ahead, but we never set any real ground rules. We never established, the issue of publication and what that means and how might we share information if we just don't make it publicly available, but to those who have a need to know. And we're really in, I think, a very, very gray spot right now. We know it's an issue, we know it's a problem, but we don't have any national or international answers on how to deal with it right now.
So again, just to be clear, so you weren't against the research in some, you were against the publishing aspect of it.
I'm basically, first of all, not clear what this research is really gaining us. And so I think that what this is really all about goes back to the very concept of a risk-benefit analysis. The benefits being in this case, does this research really give us a bright line towards an answer about what will it take for changes that must occur and then this virus becomes readily transmitted person to person? If we could know that with some certainty, you could say that's a pretty significant benefit to at least understand that. But what's really unclear to us is what changes must occur, and is it only one pathway? Could you have a series of different changes that, like a Rubik's Cube, may line up different ways on a different day? And so there is no one answer. The second thing is that there's been, I think, a number of statements made which have just absolutely not been borne out, that having this information enhances our surveillance ability, that somehow we're going to use this in sampling wild bird virus isolate or from sick humans and giving us, see, look, here's the roadmap, it's now happening. There's no evidence has made any difference in how we approach vaccine work or development of new and better vaccines. There's no evidence that made any difference in how we look at drugs and trying to treat people. And so, from a benefit standpoint, there's a real question about what is the benefit of this to society. On the other hand, the risks are real. We do have accidents happen. And in fact, the current situation today is that when we look at flu, that's the one example where we had an accidental release of a virus where, in fact, it did have international implications. Just to take a step back, pandemic influenza viruses emerged largely, we believe, out of animal populations, primarily birds and potentially swine, where there's actually reassortment, or influenza viruses get together and swap gene material between different strains that now makes that a new virus in a sense and maybe potentially transmissible by humans. And every time we have a pandemic through the 20th century, we saw that the virus that had previously been the existing seasonal flu virus, which was a remnant of the previous pandemic, disappeared and the new virus took over. So in 1918, we had an H1N1 pandemic, and nothing happened again until 1957, and H2N2 came along, and that wiped out H1N1. And in 1968, H3N2 came along and wiped out H2N2. Well, it turns out that in 1977 and 1978, the Russians and the Chinese, but we think this was clearly a Russian-related event, began working with the H1N1 virus that had disappeared in 1957. And with that, it returned, and we believe it was an accidental lab release or nothing intentional. But by the end of 1978, that virus circulated around the world again, and it came back. And that caused illness right up through the 2009 pandemic when a new H1N1 showed up. The point being is this is the one example where we actually have a laboratory isolate that got out and actually has caused many, many, many thousands and thousands of deaths over that time period that it circulated and millions and millions of illnesses. So this is not an abstract idea that flu can do this. So in this case, we're looking at the situation and saying, you know what, if this ever got out accidentally or because you published it and you now enable laboratories that wouldn't have had the ability before to do it because they didn't have a roadmap, be able to use this virus isolate in their own labs and potentially escape or potentially release, that's a whole different ballgame. This is not abstract. This is a real risk. And so you've got kind of what I call the abstract benefit package with the really very, very real risk package. And somehow we have to weigh that as a society. How do we decide that? And one of the real issues that's come up here is that this has largely been a discussion by scientists to scientists with scientists. And we believe that society deserves an opportunity to weigh in on this because the implications of any kind of release or any situation where the virus is intentionally introduced into the population has tremendous implications for society. Should society itself not be involved with some of these discussions? We think that should be the case.
You're listening to Science for the People. I'm joined by Michael Osterholm, Director of the Center for Infectious Disease Research and Policy at the University of Minnesota. And we are talking about man-made superbugs. And that's one of the arguments that I hear for this kind of research is with the growing resistance to antivirals and antibiotics, there is a need to be prepared for mutated pathogens, isn't there? Superbugs are a real threat?
Absolutely. There's no question that we need more research here. But again, this is one where we're talking about a highly transmissible infectious agent, influenza virus. It's right at the top of the transmissibility of an infectious agent. I mean, when you think about in 2009, when that new H1N1 strain emerged in Mexico and started that 2009 pandemic, within 28 days, we knew that this virus was in at least 43 different countries. That's how fast it moved. So we have no room here to be wrong. If, in fact, this virus were to get out of a lab or somebody intentionally make it and release it because they're now unable to do that, that really changes the entire risk-benefit picture. If I'm working on antibiotic resistance, for example, for a bacteria that we might commonly acquire in a hospital, there is very, 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 very limited implications for that if somebody else should ever publish that information and another lab work on it. That's not going to cause a worldwide pandemic. It's not going to even cause a local epidemic if people working on that and let's say one or two of the employees of that lab got infected with it. So the situation here is so different between these kinds of agents like the influenza virus I'm talking about and so much of the other work that we need to do. So it's not a statement about the molecular genetics of microbes and how we work with it. It's certain microbes under certain conditions that have the potential to cause literally a worldwide pandemic.
So are there the same kind of regulations around the different kinds of experiments then?
Technically, there's supposed to be in the federal government portfolio a criteria that says, look, does this have the potential to actually transmit to many, many people? Can this be a much more deadly agent because of the work you're doing? But the problem with that is it only applies to U.S.-funded research, U.S. government-funded research. And so that, in fact, the rest of the world doesn't have to comply at all. And that's one of the things that we worry about if we publish work from the U.S. government that supports this kind of activity and somebody in a foreign nation decides just to go and work on it, for which in the laboratory capability, they're limited. They have very, very little lab safety built into their program. That's an enabling behavior that now is really a serious concern. When you look at this from a standpoint of the kind of work that we need to do, yes, we do, but what it comes back to over and over again is the risk-benefit picture, and this is what I've been arguing for several years. Show me the benefits here of this kind of research, and then I will acknowledge that those benefits need to be put up against the risks. And maybe we're going to say, if the research you're giving us is going to give us the direct line to a new vaccine that could save the world if this happens, I'd say, boy, you could probably tolerate a pretty high risk. But if there is no measurable benefit that you can really demonstrate this work is doing, then your risk has to be dialed way down. And because the potential here of being wrong is so catastrophic, it's not just as simple as this bug gets out and infects a couple people in your laboratory. This is about a global pandemic. And so I think that's where we're all coming from, is saying that we have to characterize the work by what are the implications if something bad happens, and what are the benefits of doing the work that might mitigate other people from becoming infected or dying, even if this were to be just a naturally occurring event.
How do the scientists feel about the current regulations, or maybe just the policies that exist?
Well, that's like asking a question, how do men feel or women feel, or how do young feel or old feel, okay? I mean, they're very different. We have a number of scientists who are very, very concerned about this, and who almost universally don't have, in a sense, a horse in the race. They're not involved with the direct research, but there are many wise Nobel Prize laureates who have called into question this kind of work based on the publication and free-based nature which this information will be shared, and laboratory safety. The largest supporters of this are the people doing the work, and I think in some ways that's not a surprise, but at the same time, is that who should be guarding the henhouse? And so that we need to have that kind of objective third-party review. When challenged, the current researchers have yet to really put forward any kind of a logical and fact-based defense for what the benefits are for this work. And I'd be the first one if they were there to support it. You know, as I mentioned earlier, whether it's new vaccines, new drugs, anything, none of that has come from this work. Not even from our surveillance systems, how to better pick these viruses up in the community. We're often not testing viruses for months and months after they're found in patients or in animals for what we call genetic sequencing, to learn, are these changes in the virus similar to what we saw in some of our animal models? And without that, you know, that's like having a smoke alarm that only goes off once a year, whether it needs to or not.
Does this area seem to be walking the line that so many other fields that I interview about are walking, where the science is moving faster than our ability to appropriately monitor and regulate it?
Absolutely. I mean, I think this is an extension of what we're talking about. Remember, it was just a little over a decade ago that we were sequencing E. coli bacteria that would take us years to do it, millions of dollars. And now we can sequence an E. coli bacteria in one morning for literally pennies. And so we have had such a revolution in microbial genetics and genomics in general that today the tools that were once only in the hands of the most sophisticated government programs in the world are now available basically to high school students. And so one of the things we have to ask ourselves is, how much information should be out there? I mean, imagine if you could go to your hardware store and with an online internet site build a nuclear device. You know, you'd say, boy, that's really concerning. Okay, well, fortunately that doesn't happen. Well, today with microbes, we are getting closer and closer to enabling a much more amateur-like scientist group to do things with these that are really quite concerning. And as I pointed out earlier, I'm very much for the kind of research if it's really given us benefits, but I want it done safely. I want it done in places where I'm quite convinced that the virus will never escape. And I don't want that information made available to every Tom, Dick, and Harry who decides they just want to work with it. And so I think that that's the part we haven't come to grips with. It's not really about the technical aspects of the science. It's really all about how do we now move into a 21st century mindset to deal with the power of microbial genomics and what it can do and what accidents can occur because of us doing that and not understanding those implications.
You mentioned the idea earlier of sort of involving the public in these conversations. How would you see doing that?
Well, I think part of that is just beginning those discussions about how should we have oversight of this, not just in the United States, but around the world. What is the standard that we should all agree to today is acceptable or not acceptable for this kind of work? Again, if there are bad guys who want to do this kind of work, they're going to do it. Okay? And I say bad guys because they actually have nefarious intent right up front. Then there are those who you might call are the, kind of the good citizens, but they're way over their head. The kind of scientists who begin working with these viruses because they can take a publication and change the incredibly difficult process of getting these mutations, determining what they are and how to do them, and now they can just read a blueprint from a publication and do it, not really fully understanding what the implications of their work might mean. How do we bring all these different parties together in a way that we try to accomplish moving science forward, trying to better humankind because of it, but at the same time not creating these inherent risks. And we've just not had those discussions. I mean, I think this is where the public really has a right to be involved with on a global basis. You know, what risk is acceptable to me? Now, I don't mean I want every, citizen to go into somebody's laboratory. In fact, that's the last place I want them. But I think there has to be bodies where they're brought together. Our NSABB met extensively over a period of 2011 and 12 to deal with the first H5N1 things. We've been silent now for a year and a half. We've done nothing. Where are we? Why are we there? What are the international standards that we ought to all be striving towards? And I think that that's the part that many of us are frustrated with, because meanwhile, we just continue to see work like this moving forward. And again, even if the U.S. government stops or slows down how it's funding work like this, that's just fixing one of your 5 screen doors in your submarine. So what? The rest of the world has to be a party to this. In Canada, the EU, Russia, China, all these countries have to be party to this, and we just haven't seen that happen yet.
Again, something that I've seen repeatedly when we try to engage in public conversations about this kind of science or any kind of complex science, once it makes it into the media, there's a lot of hyperbole involved, and this is an area where I can see that happening.
Oh, I think you're absolutely right. Again, there's a lot of self-interest here. For example, I mentioned earlier to you that the groups that really have been promoting this kind of research are largely the groups who are funded to do it. Well, that makes sense. But then can they be seen as the objective party to come forward and say, oh, by the way, these are all the benefits from this? We need a neutral third party that are expert to be able to say, no, wait a minute, this isn't really benefits at all. And so to us, I think that's those scientists who are concerned about this. Remember, I do a lot of work in influenza. So I'm one of those people that kind of cross the bridge in the fact that I am concerned about influenza. It's a very serious potential public health catastrophe waiting to happen, particularly when if the next pandemic turns out to be a virus that's quite lethal. So I don't want to miss the opportunity to have better drugs or vaccines, etc. But I'm the first to also say, don't tell us that this is a great benefit when it's not. And this is where we just have to keep coming back to what I think is a common sense approach that any citizen off the street corner should agree with is, what are the risks? What are the benefits? Let's have an objective measure of the risk. Let's have an objective measure of the benefits, and go from there. And then decide how we're going to work on this. And if you can't define really meaningful benefits, then what the hell are we even doing talking about risk for? Any risk will be too big. And that's, I think, where we're at today.
Michael, thanks very much for being here.
Thank you.
If you're looking for Michael Osterholm, Director of the Center for Infectious Disease Research and Policy at the University of Minnesota, you can find him at the link on our site at scienceforthepeople.ca.
Science for the People is a weekly radio show exploring everyday life from a scientific perspective. To find out where Science for the People airs near you, or to listen to past episodes, check out our website at scienceforthepeople.ca. You'll also find links to connect with us on Facebook, Twitter, and Google+, or to subscribe to the podcast in iTunes. And now, back to the show.
Welcome back to Science for the People. I'm Desiree Schell, and I'm joined by Associate Professor of Biology at Western Washington University, Benjamin Miner. Ben researches how the environment affects marine organisms, and his lab has done some work around what we're here to talk about today, namely starfish wasting syndrome. Good to have you here, Ben.
Thank you.
Now, when did we have the first inkling that there might be a plague among starfish?
Well, the syndrome this year was first detected in the summer, and there was research biologists out on the Olympic Peninsula, in particular Dr. Steve Radkin with the Olympic National Park, surveys a lot of their intertidal sites and for a variety of organisms, but sea stars in general. And he was noticing that there was, at one of his sites in particular, a number of starfish that didn't look quite right. They had lesions, some of them were missing arms, and basically alerted a number of the other groups that he works in collaboration with all along the coast that he was seeing something that looked a little bit different. About that same time, a little bit later, there was some pictures taken from a diver the Vancouver area that was showing that there was a number of dead stars, and they were basically kind of piles of goo, which is classic, wasting syndrome look to sea stars once they've died. And then posted that on a blog that was basically went out to people interested in echinoderms. And then about that time, people started getting interested and started looking more seriously around detecting it in several other places. So that was kind of the beginning. Now, that being said, starfish have been identified having similar symptoms in the past. So this wasn't the first year that we've seen starfish dying of these symptoms.
Well, how many starfish are we talking about exactly? How many have we lost to it?
The estimates are extremely difficult because we've only sampled, a subset of sites. But I would bracket it somewhere in the tens of thousands to probably millions, somewhere within that range.
What percentage of the population would that be, the total population?
For certain species, it'll depend because this is across quite a few species that appears to be affecting. I would say that it's probably less than 25% of the population, probably somewhere between 5 and 25%, but that's an extremely rough ballpark guess.
So have we only seen this disease or syndrome in North America?
Yes, it's only been reported, as far as I know of, in North America. However, it has been reported on the East Coast and the West Coast.
Before we actually start talking about how the syndrome works, maybe let's set the stage. And can you tell me a bit about the normal physiology of a starfish?
Typically, starfish they're covered by skin. They have an internal skeleton, just like we do. In fact, they're in a group that's related to us, relatively closely related to us among animals. One of the things that's interesting about them is they have what's called a water vascular system. And so their circulation is basically through a series of tubing within their body. Many people who've seen starfish will know that they have these sucker tube feet that they use to walk. They also use those to sense the environment. They also use those to breathe, too. So this water vascular system is kind of a multi-purpose system that does a variety of things. Within the fluid that circulates within the water vascular system, some of it is drawn from the external environment through kind of what's called a sieve plate, and then and that can allow for the pressure to change within the system. But they also release cells into this fluid that act as immune defenses. So when foreign objects get in there, these cells will glom on to foreign bodies and oftentimes kind of encapsulate them and then help defend the individuals.
What does the disease do to them exactly? How does it start?
So the disease, it's not actually all that well understood yet exactly what happens. Certainly the symptoms that people have identified, and they vary among species a little bit, but basically what happens is the individuals will lose kind of turgor, so they'll lose pressure within their bodies. That may not necessarily be due to kind of the lack of functioning of this water vascular system. It may just be a reduction in the rigidity of the body wall, but they get a bit deflated looking, a bit flaccid. They'll have lesions typically in the armpits of the individuals, but they can occur in other places. And then those lesions will spread. Oftentimes then the body wall will rupture. Some of the internal organs may spill out. The arms oftentimes will rip away from the body. And then about at that point, the individual succumbs to death. So what is believed to happen is that at that particular point, the immune system has been severely compromised previously, and bacteria have started to break down a lot of the tissues. And so the individual dies. The bacteria have already started to break down the tissues, and then just in a short amount of time, the individual basically deteriorates.
So, I do have to mention this at this point because I like to terrify myself. This is potentially the most horrific thing I've ever heard. After the arms fall off, the arms may continue to crawl around for a while?
They do, and at least one of the species that seems to be very susceptible, it will eat just about anything. So we've seen it in the lab, but people see it in the field too. The arms will rip away from one individual, and another individual will come across and rapidly consume those arms. And so certainly it's quite horrific to watch.
You have a strange job, sir.
Yeah.
This is Science for the People, and I'm talking about starfish wasting syndrome with Ben Miner, Associate Professor of Biology at Western Washington University. Okay, so you mentioned that we don't know very much about it. So let's start with what we do know. I'm alternating between calling it a disease and a syndrome because I've seen articles use both terms. So is there a distinction?
It was initially termed a syndrome just because it was a set of symptoms that people were observing. My colleagues that are actually disease biologists that study disease all the time say that there isn't really any need to distinguish between those terms. That a disease can be anything from a pathogen to something that's just an environmental pollutant, for example, that's affecting individuals. So it's been called a syndrome in the past, and I think people have been careful to use that term just to not elicit the basic idea initially that it is a pathogen.
Well, and it affects all different species of starfish, correct?
Well, it doesn't affect all species. Well, at least not that we know of. There are, I would say, about 10 species that appears to affect, but for example, in the area near me, the Salish Sea, the inland waters of Washington, there's, I don't know, between 20 and 30 species of sea star here. So certainly there's a number of species that I see healthy all the time, even at infected sites.
And can it be passed to non-starfish?
Yeah, as of right now, there is no evidence that the same syndrome is affecting things that are not starfish.
And why don't we know what causes it?
The issues with disease, especially for something like a pathogen, is that once the individuals become sick, they can contract a variety of different diseases, basically different things that can damage their tissues or cause them to be sick. So, for example, a good analogy is kind of the human AIDS virus, where initially people were dying of, kind of a variety of symptoms. It wasn't actually the virus per se that was killing them. That was just suppressing their immune system, and then they were dying of a variety of other illnesses. So, that could be the flu, or that could be some type of bacterial infection. And so when you look at those cases initially, it doesn't appear that there's anything in common necessarily with exactly, how they're what's causing them to die. And that's similar to what you have with a lot of diseases, that you can identify something that's consistent, but it's not clear whether that's the root cause that's initially making them sick, or that's just the secondary infection that's causing them to die after they've gotten sick and had their immune system suppressed. And we can envision for starfish a whole variety of environmental insults that might be affecting them, from pollutants, to increased temperatures, increased or decreased pH levels, and changes in salinity. All of those things likely stress the individuals out and may in some way contribute. And so it's difficult to pinpoint, because sometimes the things that actually made them sick initially may be very hard to detect after the individuals have subsequently gotten additional infections or gotten sick from other things.
Well, and there's been a litany of potential causes that people have put out there. You mentioned some of them. Can you go into a bit more detail?
Yeah, I would say the general hypotheses that have been stated are a wide variety of environmental contaminants that are basically products of man-made things, and to just natural fluctuations in the environment, to pathogens, other organisms that are infecting them. All of those have certainly been suggested as possible causes. The reality is that again, the complexities of ocean currents and the geographic separation of where we've seen the major die-offs makes it very difficult to come up with one particular hypothesis that fits all these scenarios well. And so it's very likely that the actual explanation will turn out to be much more complex. It's not just this thing, but it's these three things here, and it's these other three things here, in this other area possibly. And so So basically, that's, people are working trying to gather evidence, related to all of these hypotheses, and certainly given the relatively short amount of time since it's shown up, people are just kind of getting underway.
Well, I originally read about the syndrome on Facebook because someone posted an article attributing it to Fukushima. So is there any truth to that?
Certainly, the location where we've seen the major die-offs doesn't seem consistent at all with that hypothesis. Obviously, you would expect from the way it would come into the inland waters, for example, would be that it would hit the outer coast first. And yes, we did initially observe at one particular site on the outer coast, but there's lots of other sites essentially wrapping all around the outer coast of Washington and in through the Strait of Juan de Fuca and in through the San Juans that all are still very healthy populations. And we would have expected to kind of see it initially come through that area, wiping out those areas, and then finally kind of getting into the more protected waters. However, we see almost the exact opposite pattern. The major die-offs have been occurring in these more kind of protected waters in the Vancouver area, the Seattle area, the Monterey area, and down in Southern California. The other thing that also suggests that it's not that is that the basic spread at which we expect the currents to move shouldn't be bringing any radiation down into the Southern California waters yet, but yet we're seeing the disease down there. So that also suggests that it's the case. And then when people have basically looked at levels of radiation within the waters associated, they haven't seen elevated radiation either. In addition to that, the major type of radiation that's coming across is the one that's likely to be most hazardous to marine organisms is going to be cesium and 137, and that will basically mimic potassium which is a really important molecule or chemical in the physiology of a lot of animals. There's something called the sodium-potassium pump that's essential for basically generating energy among animals, and all animals have this, and we would expect then the cesium to be affecting a wide variety of animals, not just focusing in on sea stars. So basically all of those things suggest that that's not the direct cause. Now, certainly, it doesn't rule out the possibility that it is in some way contributing, potentially reducing the immune system of individuals and potentially playing some more minor role within the syndrome.
Well, what about the idea that climate change may have something to do with that?
Again, the pattern that we would expect from, climate change could be broad spread in what we're seeing, but the die-offs are kind of localized in certain areas. And so again, it's kind of hard to envision anything specific that would have allowed for the die-offs in these localized areas that would be kind of the result of climate change. However, again, that doesn't really rule out the possibility that it's, again, playing some role, but not the direct role, and maybe weakening individuals, making them more susceptible to other insults.
Okay, so for comparison's sake, how often do we see outbreaks like this among marine life? And I don't mean wasting diseases necessarily, but mysterious plagues that kill off large numbers of a species.
Yeah, well, we've seen diseases in kind of large-scale diseases, at least. In marine systems, a handful of times, certainly, that have been well documented. In general, the problem that I'd say makes it difficult to estimate kind of how common, diseases within marine systems is that a lot of systems we don't know exactly, they're not well enough studied that we know kind of this is, what organisms do normally, and these are sick individuals. So mostly diseases manifest themselves and catch the attention of marine biologists when they're large die-offs of certain individuals, and oftentimes particular species that are very obvious and people have, seen. So, for example, down in the Caribbean, there was the die-off of sea urchins down there. Lots of people, dive around there, and they noticed kind of the distribution of sea urchins over time, and then when they started to die, it was very noticeable. And the subsequent changes to the environment were also very noticeable too. So again, starfish fit that basic mold. They're very obvious, relatively large among marine organisms, and don't move around a ton too. So when they start disappearing, it's likely that they're dying, not just relocating themselves. So those are, I think, typically the types of organisms that people have noticed. Marine mammals get studied fairly well, and their physiology is more similar to ours. So we have a lot of tools to test the health of people, which can also be utilized to test the health of marine mammals. So those are basically the groups that oftentimes get targeted and noticed. We've seen disease certainly throughout all the oceans for particular groups. But there's only really just a handful of cases that are really well studied and well known among marine species.
For those people that don't live near coastlines or possibly have no innate love for starfish, should this disease matter to them?
Members of ecosystems oftentimes affect how other organisms interact, can affect the products that those ecosystems are able to produce for humans or the aesthetics of those particular habitats. So in some general sense, the care, I would say, in almost any situation is that when we see large-scale die-offs of organisms, that those communities might change in ways that are going to be detrimental. It's also imaginable that they could change in ways that could be beneficial to people too. For example, sea stars might eat a particular species that people also like to eat. So when sea stars disappear, there's more of those species for individuals for people to harvest and consume. So again, the outcomes of what happens in these communities can be difficult to predict because of the number of interactions with other species. But in general, I would say that as ecosystems are radically modified in the very short term, it makes it just in general difficult to understand and predict what those systems are going to be like in the future. And a lot of our general understanding of what we're going to be able to harvest from the ocean depends on that understanding. So when we start perturbing the ecosystems, it makes those predictions less reliable, and that can affect your predictions of how many crabs we're able to take out of the Puget Sound, or how many fish we're able to harvest and ship. And oftentimes those products are shipped everywhere, even to inland locations.
Ben, thanks very much for being here.
Oh, no problem. Thank you.
And we've linked to Ben Miner, Associate Professor of Biology at Western Washington University, on our website at scienceforthepeople.ca.
On the next episode of Science for the People, we're peering out into the black to learn about deepest space and our own night sky. We'll talk to bat astronomer Phil Plait about recent measurements of gravity waves and what they tell us about the birth of the universe. We'll speak to journalist and essayist Paul Bogard about his book The End of Night, Searching for Natural Darkness in an Age of Artificial Light. And noisy astronomer Nicole Gallucci tells us about a project using citizen science to map the surface of the moon. That's next week on Science for the People, on your local radio station, or online at scienceforthepeople.ca.
I'm Rochelle Saunders. I'm here with Ross Lockwood, who is finishing his last year of his PhD in condensed matter physics at the University of Alberta. Ross has a passion for scientific outreach in astronomy and regularly participates in public and educational initiatives, communicates his understanding of science as a guest lecturer at the University of Alberta, and as a writer for the University of Alberta's Science Contours magazine. He has been selected as one of the participants of the upcoming High Seas Mars simulation mission, which begins on March 28th. And he's here today to talk about his upcoming pseudo-space adventure. Ross, welcome to the show.
Hi, Rochelle. Thanks for having me tonight.
So what is the High Seas project?
So High Seas stands for the Hawaii Space Exploration, Analog, and Simulation. And what that basically is a Martian simulation of a habitat. And we'll be spending 120 days inside of this habitat doing various projects and tests for certain systems, with the end goal and the principal investigation being our psychological health.
You're not actually necessarily simulating life on Mars from the standpoint of its physical hardships, but more its mental hardships?
That's the goal of this study. So the first High Seas mission was actually to investigate the dietary hardships that astronauts might face on a long-duration space mission, and what ingredients they could take and how varied their diet could be just based on those shelf-stable ingredients. So there's a series of 3 missions that are longer and longer durations that will be investigating the mental well-being.
What kinds of data is this study looking to uncover?
The primary objective is to establish how to construct teams for long-duration missions. And they're going to be doing things like evaluating our short-term and long-term cohesion so that they can improve astronaut selection for long-duration missions. But in addition to that primary study, we'll also be testing a number of other things throughout our stay, from antimicrobial clothing to soil science during our spacewalks, and extending the food study.
So how many people are participating in this mission?
So on the inside of the dome, there will be 6 people total, myself and 5 others. And we have quite a large support crew that will be monitoring us on the outside. We really only are in contact with 2 people, the principal investigator and her postdoc. And the entire psychological team, as they say, is behind the curtain.
So this really is to try and get as close to possible as the real working conditions of a long space voyage, as in very few people to interact with.
Yeah, that's exactly right. There's typically one Capcom that communicates with the crew of any long-duration space mission. So, for example, on the International Space Station, there is one person on the ground that serves as the point of contact between mission control and the astronauts on board. So we'll be trying as much as possible to simulate that. But additionally, we have other forms of communication, specifically email, that we'll be able to send freely back and forth as long as it goes through a 20-minute time delay on the outbound and 20-minute time delay back on the inbound for a 40-minute round trip delay total. And that's to simulate the speed of light distance between Mars and Earth at their furthest.
I had heard that you guys would be able to communicate via email, but the fact that there's a 40-minute time delay means that there's no sort of chatting via email that you can really do.
Yeah, and I think that's one of the golden rules that we can't break on this study. We've been trying to figure out ways that we can do outreach, either all by email or by setting up systems where we can pass an email message out that gets pushed to Twitter and Facebook and all the other social media we plan on using. But that's actually one of the bigger challenges, is how we're going to be communicating with the outside world.
So how will the psychological team be monitoring you guys during your stay?
So from what I understand, we'll be given a specific tablet with a bunch of psychological tests and surveys. And on a daily basis, we'll be filling out those tests and surveys to evaluate certain psychological factors. Now, of course, I don't know very much about that right now, because if I did, that would affect the outcome of the study. So instead of telling us exactly what to expect, they just tell us the method in which we're going to be being tested.
You mentioned spacewalks before. So you actually get to go and simulate spacewalks as well as the sort of day-to-day living conditions?
That's right. And this is one of the activities that I'm most excited for. When we do spacewalk, again, we're simulating everything we can as far as conditions on Mars might be. So we can't simulate things like Martian gravity or the Martian atmosphere, or even the fact that astronauts on Mars would be using pressurized spacesuits. But we are required to maintain a pressure seal on our suit. So, that means that we can't take our gloves off when we're outside or our helmet off. And for all intent and purpose, we're going to be actually walking on the surface of Mars and doing geological work that astronauts on Mars might one day do.
This is really fascinating. And I suppose this is one of the reasons that the simulation site was chosen where it was, which is in Hawaii, correct?
Yeah, that's right. We're on the slopes of the Mauna Loa volcano, and that's similar to some of the geological sites that are present on Mars. So there's these large shield volcanoes on Mars, and the stuff that we learn here would be applicable to the kinds of things we could be expected to do when we send humans to Mars.
Okay, so living arrangements. Obviously, if you're simulating living on Mars, we're probably not talking a lot of space.
Yes, and this is another one of those interesting things that it's going to have a big effect on the actual psychological impact on the crew members. So the 6 of us will be living inside of a geodesic dome that's 36 feet in diameter, and that gives us about 1,000 square feet of living space on the main floor, where the kitchen, the workspace, and the experimental areas are, and a little under 600 square feet on the second floor, where our bedrooms are. So our bedrooms basically consist of a single bed, one nightstand, and they taper down from the widest part of from the longest part of the bed to the door.
Wow, that's not a lot of space for 6 people.
Yeah, you're right. Although there is one really interesting thing about the living conditions that I don't think many people consider, and it's kind of the extremes of closeness and loneliness that we have. So with the delay, we're really simulating that would be millions of kilometers away from Earth, but inside the dome, we're simulating what it would be like to live with 6 other people in extremely close quarters.
It's sort of like getting assigned a bunch of roommates in college or university, where you kind of don't get to choose the people you live with. So there could be some interesting personality conflicts, or maybe everybody will get along really well. That's really fascinating, because I guess on a real long-term space voyage, the individuals going don't necessarily get a veto on somebody else.
Exactly right. And one of the main conditions we've been told that we've been selected for was our astronaut-like abilities. And that really doesn't say anything as far as personality is concerned, other than some fortitude for work and some experience with science and maybe some psychological background. But as far as the actual personalities are concerned, we've really only had a few dozen hours now on Skype where we've been able to get to know each other.
So I'm curious, the power supply, are you guys plugged into the grid, or is everything Mars-like?
No, we learned this week that we'll be using a new power system, which consists of an array of solar panels and a bank of batteries. Now, they've told me that that's giving us between 8 and 10 kilowatt hours of power per day. And as far as power requirements go inside the dome, if all of our experiments are running and all of our kitchen supplies are running, we'll actually run out of power before the end of the day. So power management is going to be a big thing. But with that being said, we do have a backup system, and this is a fuel cell. So if we do run into the red with the solar panels and battery, we'll be able to kick on that backup system and use that for low power requirements.
So what do you think the most challenging part of the simulation will be on the other side of it?
For me, it's predicting what the impact will be on my own personality over the duration of it. This is one of the things where, in the 2 months that I've known I'd be participating on this, I've tried to kind of build up a list of activities and projects that I can work on inside. But I really have no idea if I'm going to be able to do those in the time I've got allotted, or if I'm going to be doing some other things. And I really have no idea how that will impact my personality. So when I look 120 days into the future from the day I step in there, I can't really predict the person that I'm going to be stepping back out again.
Well, it sounds like a really interesting adventure, and I wish you and your team a ton of luck, and hopefully we'll get one of you guys back on the show after it's all finished to talk about the actual experience.
I'd love to be back on the show for sure. Thank you so much.
If you want to keep tabs on Ross Lockwood and the High Seas mission, you can follow his blog at spincrisis.net, where he'll be writing about his experiences. You can also follow him on his public Facebook and public Twitter feed. Obviously, there'll be a 40-minute time delay on those, but we'll provide all of those links in the show notes. And you can find those show notes on our website at scienceforthepeople.ca.
Science for the People is recorded in Edmonton, Alberta. The show is produced by Rochelle Saunders. Ryan Bromsgrove is our promotions manager. Our social media manager is Chelsea Butler. K.O. Myers updates our website. Helen Quivalon is our publishing liaison. Ed Haynes is our guest coordinator. We get research help from Josh Witten. The show is edited by K.O. Myers and Ryan Bromsgrove. Our theme song was written and recorded by Fractal Pattern, and its title is Binary Consequence. The show is hosted by Rochelle Saunders, Marie Claire Shanahan, and me, Desiree Schell.